partial reprogramming can epigenetically prevent or maybe even reverse Alzheimer's symptoms. The beauty of epigenetic changes is that they're reversible. Partial reprogramming can prolong lifespan in both progeric and wild-type mice. Aging might be reversed. Partial reprogramming can even induce neurogenesis in adult mice. Thank you. Thanks for coming. I don't think I've ever done a talk in sunglasses before, so this is a first for me. But yeah, wanted to tell you about partial reprogramming for the brain, in particular Alzheimer's. That's what we're doing at Youth Bio, and I've been working on translating partial reprogramming for almost a decade. I was just reminiscing. It's been so long, and unfortunately, it's been a slow journey, but hopefully we'll get there soon enough. And yeah, thanks to Daniel, you're all experts in reprogramming now, so I don't need to go into the basics. I can just kind of pick up where he left off and talk about just very briefly how partial reprogramming works, and then just go into more details about the talk on the brain, actually how we use partial reprogramming for brain diseases. Just very briefly, to summarize why I'm so excited about partial reprogramming, it seems that reprogramming can rejuvenate so many different cell types, has already been demonstrated to do so, and so the potential of reprogramming to revolutionize medicine is immense. And this is what got me excited about partial reprogramming back in like 2017. And since then also got a lot of people with a lot of money excited, like Jeff Bezos, Sam Altman, Brian Armstrong, and this led to many partial reprogramming companies having been started by these people. And why, again, is reprogramming so great? Because during the reprogramming process, a lot of the hallmarks of aging, in fact, all of the cellular hallmarks of aging, and there's some extracellular hallmarks of aging, but all of the intracellular hallmarks of aging are rejuvenated by the reprogramming process. So while things go bad in this direction with aging, if you do reprogramming, things are ameliorated, and all of the hallmarks of aging demonstrated on this slide are ameliorated by the reprogramming process. And this is the full reprogramming process where the cell is taking all the way back to the pluripotency. But of course, in the field of longevity, once we realized that this happens with reprogramming, the question was, can we harness this rejuvenating potential of reprogramming and use it in vivo? And the study that provided the first yes to this question is the seminal Ocampo et al. Study in 2016 from the group of Juan Carlos Espiso Belmonte out of the Salk Institute, where they have demonstrated that if you use reprogramming for short durations, 2 days on, 5 days off, and you do this repeatedly on a weekly basis, you can extend lifespan of progeric mice by up to 50%. And this was a great result that got everyone excited and essentially started the partial reprogramming field. And since then, not only progeric mice, not only transgenic mice, but normal, regular, wild-type mice have been shown to extend lifespan by partial reprogramming. This is a study that Daniel's already referenced by Rejuvenate Bio, where they took really old mice. These mice were like 2.5 years old, which is like really old mice who barely live, you know, like 2.5 years. But they did a partial reprogramming gene therapy, injected these mice at this old age, and this doubled the remaining lifespan. But overall, the overall lifespan wasn't that exciting in terms of increase, about 8%. But then another study has also been done in wild-type mice. It has shown a 12% increase. But more importantly to this talk, and for our work at Youth Bio, in terms of diseases, partial reprogramming has been shown to be effective in various disease models. And in particular, Alzheimer's, which is the focus of Youth Bio and this talk, there's already a published study showing that if you deliver partial reprogramming gene therapy into the brain, into the hippocampus in particular, and then you activate this therapy, you can prevent the hallmarks of Alzheimer's. And this is one of the reasons why I'm so excited about partial reprogramming. And I'll talk in more details later on, but this is kind of like the preview on why reprogramming is so interesting and so exciting. And again, picking up where Daniel has left off, how does partial reprogramming work? And the answer to the rejuvenating aspects of partial reprogramming are still unclear. We're not yet sure how exactly the magic happens, but we can share some observations and then kind of speculate based on those observations what might be happening and how rejuvenation might be achieved with the reprogramming. And of course, reprogramming is done by Yamanaka factors, and I'm sure most people know about the role of Yamanaka factors in terms of stem cells and preserving the stemness of these embryonic stem cells. But what is sometimes not known about Yamanaka factors is that these factors are also factors that trigger this maternal to zygotic transition during embryogenesis. And this starts at the blastula stage and goes on until gastrula stage. And why this is important? This is important because there's some observations that gastrula stage or gastrulation is actually the ground zero of at least epigenetic aging, also some indications transcriptomic aging. So basically, there is this active rejuvenating process that happens after fertilization that minimizes epigenetic age and essentially rejuvenates organisms during embryogenesis that takes its place during gastrulation. And there is also data from single cell resolution and data from frogs that show that this happens. And the idea then that ties back Yamanaka factors to this process is that Yamanaka factors during reprogramming, they could be initiating the same rejuvenating subprogram that is active during embryogenesis, and the subprogram during embryogenesis ensures that our age is reset when we're a new baby. But also during full reprogramming and during partial reprogramming, the initial stages of reprogramming could be also activating this rejuvenating subprogram. But this was speculation. This was just kind of the hypothesis that I have. But moving to what we actually observe, while we might know how exactly the magic happens, we know that the magic does happen because we see epigenetic, transcriptomic, metabolomic, proteomic rejuvenation with partial reprogramming. This is one study showing that partial reprogramming rejuvenates the transcriptome. Another study also showing that partial reprogramming restores youthful gene expression. And so these are already empirical observations that we're trying to now harness and translate into therapy. So we might not know exactly how things work, but we know that they do work to some degree, and so we're trying to make it work for therapies that we can use in humans. And in terms of also, I mentioned metabolomic, but at the tissue level, partial reprogramming has also been demonstrated to essentially rejuvenate cells or make them more functional, bring them to a more functional state, because some people are like, well, what exactly does it mean to be more youthful? What we really mean is more functional. Now, moving on to other experimental results that have come out since the 2016 Ocampo paper, I just want to highlight some of the studies that have been done since that I find particularly compelling. One of the first studies that was done was essentially replication of Ocampo's results. This was done from INSERM, Jean-Marc Lemaitres' team in France. They essentially replicated the findings of Ocampo et al. And showed that even a single induction of partial reprogramming can prolong lifespan in both progeric and wild-type mice. This is a result that a lot of people might know from David Sinclair's lab, where they showed that you can regenerate some neuronal connections and also restore vision in this nerve crush model of vision loss. And this is what led to the establishment of LiveBio, the company of David Sinclair, which is now gearing up to go into humans very soon. This is a study that's showing partial reprogramming can improve muscle regeneration. I won't go into too much detail in the interest of time, but there's several papers that showing various aspects of partial reprogramming having therapeutic benefits for wound healing, for for spinal regeneration. And also, this is a study out of Belmonte lab, subsequent study to the Ocampo study, showing that even long-term partial reprogramming is safe and provides therapeutic benefits. So this is a slide summarizing safety, and this slide summarizes therapeutic benefits of partial reprogramming. And I already mentioned the wild-type mice because a lot of people were kind of waiting for results in wild-type mice because initial results were in progeric animals and transgenic animals. And I mentioned this is a study in normal mice, really old mice, in which partial reprogramming doubled the remaining lifespan. And then the subsequent study also done in really old mice, but in a way that the formulation was geared towards senescent or pre-senescent cells. That partial reprogramming therapy, when administered to, again, pretty old mice, 18 months old mice, a year and a half, this extended their lifespan by like 12%. And there's also some yet unpublished results, again, with promising efficacy, promising signals of efficacy that, again, I won't dive too deeply into. Let's go into brain rejuvenation in the interest of time, because there's still a lot of material that I'd like to share with you. One of the first studies was done by Manuel Serrano's group in Spain, showing that partial reprogramming can improve memory in wild-type mice. And the recent paper out of Stanford, Anne Brunet's team, showed that partial reprogramming can even induce neurogenesis in adult mice. And adult neurogenesis has been a very controversial topic of a lot of research. Some people saying adult neurogenesis is impossible. Well, at least within partial reprogramming, it seems that adult neurogenesis is possible if induced by partial reprogramming. Another study out of Argentina showing that partial reprogramming can have beneficial effects in the brain in old rats, show that you can improve cognitive function in old rats when you inject this therapy directly into the hippocampus of old rats. And this is the same modality that we're using in Youth Bio in the context of Alzheimer's and age-related cognitive decline. So it's nice to see other groups also obtaining essentially similar positive results. And in that study, they not only saw positive improvements in cognitive behavior, in tests of cognition like this maze, but also they saw reduction in epigenetic age of the treated rats, which is another additional data points to the efficacy of partial reprogramming or rejuvenation observed during partial reprogramming. And this is a study that we did jointly with the Ocampo lab, so it's a Youth Bio study together with the Ocampo, where we tried to see if we can ameliorate age-related cognitive decline in normal mice. So these were, again, old animals, like 17 months old mice, into which we injected directly into the brain, into the hippocampus, our therapy, and we saw improvement in memory and learning. Again, this was a very advanced age of these mice. We went up testing up to 26 months of age. Now on to Alzheimer's, the main focus of this talk and my personal passion, because this is something that runs in my family, so I'm a little biased in terms of thinking why Alzheimer's is a really good indication in general for longevity therapies or for partial reprogramming in particular. But I think even without my kind of personal views, I think there's good reason to think that Alzheimer's is a good candidate, because there's a strong signal of epigenetic etiology to Alzheimer's. And also the way that partial reprogramming works, it works at the level of epigenetics first and foremost. So there is I think, good reason to believe that partial reprogramming can epigenetically prevent or maybe even reverse Alzheimer's symptoms, because the beauty of epigenetic changes is that they're reversible. Epigenetics can go in either direction. And one of the first studies that showed that Alzheimer's symptoms can be epigenetically prevented in an Alzheimer's mouse model was this study that basically showed that this is the kind of proof of concept. And another study that showed that you can even reverse Alzheimer's symptoms using epigenetic alterations was a study in 2019. And they showed that, not using partial reprogramming, but using other mechanisms of epigenetic modulation, you can reverse symptoms of Alzheimer's disease in this particular mouse models. And finally, there's now a published study showing that with partial reprogramming, with targeting the neurons in the hippocampus, which is the same modality that we've used at Youth Bio, but we haven't yet published our results. But this group has published their results. University of Barcelona led the study, where they have shown that using partial reprogramming in an intermittent fashion, neuron-specific partial reprogramming in the hippocampus, you can prevent symptoms of Alzheimer's disease. And not only can you prevent the molecular biomarkers of Alzheimer's, like levels of amyloid beta in the brain, but also cognitive performance on various tests assessing cognitive function, that you can, the treated mice did better than the untreated mice, and so you can claim that you are preventing the functional decline, the cognitive functional decline of at least mice in this particular model. And so, of course, now the million-dollar question, or even billion-dollar question, is how do you translate this into therapies that we can all use as humans? And in general, of course, in the longevity space, first, before you can actually have a therapy that can have systemic effects, you have to demonstrate its impact in a particular disease model so that you can get on the market, and then maybe you can label expand. And so we think with our approach, we're starting with the brain. We think if we can show meaningful therapeutic benefit in CNS diseases like dementia, Alzheimer's, first and foremost, then we can iterate and expand into other organs, other indications, and ultimately we want to have a combination therapy that healthy people can use as prevention and potentially rejuvenation. But starting at like stage 1, our therapy is using Yamanaka factors delivered directly to the brain, and this is an inducible Yamanaka factor therapy that we deliver to the brain once. We deliver to the hippocampus directly with an injection to the hippocampus, bilateral injection to the hippocampi, and then we activate OSKM on a periodic basis as partial reprogramming currently needs to be done. It needs to be activated on a weekly, at least in mice, or potentially bi-weekly basis in primates. And the clinical trial we envision is in patients with either mild cognitive impairment or already early diagnosis of Alzheimer's, potentially with a subgroup that are most at risk of Alzheimer's, people who are positive for the ApoE4 allele, as I am. But in terms of precedent, there's actually a lot of precedent in delivery of gene therapies to the brain. So because some people are a little bit scared of the brain as an organ for gene therapy because they think delivery is so hard. But actually there's a lot of precedent in the neurodegeneration space of just delivering directly to the affected area of the brain. And Parkinson's, of course, leads the way there because there's already a lot of therapies that have been, gene therapies that have been delivered to the substantia nigra or the motor cortex, and trying to see if there is positive effects from those therapies. And moreover, there's even lentiviral therapies, because lentiviruses are another thing that some people are a little cautious of. But there's actually 2 different therapies already that have been tried in humans, in human clinical trials, in Parkinson's patients with lentiviruses, the first of which is Pro7 or Pro7, if you're from France, which has been already studied for over a decade and has been demonstrating a very safe profile of administration, even with the direct injection to the brain. And the second therapy, lentiviral therapy, in the context of Parkinson's is AXO-LentiPD, which is essentially a successor to Pro7, but also it was well tolerated in its human clinical trials. And in addition to lentiviruses used in the CNS, lentiviruses have actually been already approved. There's a couple of therapies, lentiviral therapies that have been approved by the FDA, and also a few others that are on the way and considered in clinical trials. In addition to the Parkinson's trials that I mentioned, there's also other trials that are on the way of lentiviral therapies, and Oxford Biomedica leads the way in that department in specializing in human gene therapies that use lentiviruses. Switching gears to Alzheimer's, there's actually also precedent in Alzheimer's of direct delivery of gene therapies to the brain, to the region essentially near the hippocampus, where in 2013 they delivered this AAV gene therapy to the brain of Alzheimer's patients. And they followed up 7 years later and showed that this therapy was well tolerated, and moreover, the expression of the gene that they delivered was stable throughout all these 7 years, which is a good indication that we can count on our cargo to also be stably expressed for years and years if we deliver it to our patients. And another example, not a gene therapy, but a cell therapy, but still the same level, same route of administration, direct delivery to the hippocampus of the cell therapy in Alzheimer's patients. So basically, all I'm trying to say is there's good precedent already for this type of therapy, this type of modality being delivered to the brains of human patients. But ultimately, our goal, of course, is to have non-invasive delivery, and one particular potential way we can do this is using ultrasound-guided delivery, and this modality has also been explored for many, many years in various disease models, and actually even tried in human patients in Toronto, actually, our hometown, as Nathan mentioned, in pediatric patients with glioblastoma. It was successfully trialed there. So as kind of the future of these therapies, there's also potential for non-invasive delivery of these therapies to any region we want to target in the brain. And also, in addition to these lentiviruses being used in humans, there's some precedent of partial reprogramming actually already being used in humans. Of course, this is with an asterisk because this is ex vivo therapy. These are T cells from Lyell Immunopharma that have been rejuvenated with partial reprogramming, but they were rejuvenated ex vivo, but then they were, of course, used on the patients in these 2 clinical trials for the 2 of these products. But so you could say that there's already precedent of human use of partial reprogramming. And also, we're well on the way as a field of having therapy being tried, in vivo partial reprogramming therapy being tried in human for the first time. This is Live Bio, as I mentioned. They're aiming to have an ophthalmologic indication based on the research of restoring vision in their nerve crush model. They then progressed and have focused on glaucoma and this NION indication, which is essentially eye stroke. And eye stroke is the indication in which they demonstrated in primates, non-human primates already, that this is a safe and effective modality, and they're gearing up to a human clinical trial within this year. Initially, I think their plans were like mid 2025 to start a human clinical trial, and maybe they have already started. So there's, I think very soon we'll see the first patients using partial reprogramming. And also another company, TurnBio, is also well on its way to human clinical trials. They had good positive feedback from the FDA at the Interact meeting for their therapy in skin wound healing conditions. And yeah, we're at also Youth Bio, we're on our way to talk to the FDA, have our own Interact meeting, hopefully to get their feedback for our human clinical trial in Alzheimer's patients. With, if everything goes well, we expect to see it within 2 to 3 years, of course, pending the required funding, which is yeah, everyone's current challenge with fundraising. Just to close, I think future directions for partial reprogramming, where we see the field going, is moving away, eventually moving away from Yamanaka factors. But I think while some companies are focusing on that, like Shift, New Limit, and others are focusing on finding null factors, I think we can still try to translate existing knowledge of using Yamanaka factors, because we know these things work and we know they're relatively safe. So for the first generation of therapies, I think it's good for the patients to have at least something that can provide a meaningful therapeutic difference, at least in the context of Alzheimer's, where the clinical need is huge and there's millions of patients. But of course, ultimately, I think we'll need to be moving to cell type specific cocktails with novel factors that target a specific cell population in a targeted way that are safer than the current Yamanaka factors can provide. And yeah, with that, just want to close with this very inspiring quote from Juan Carlos Espiso Belmonte, the leader of the group of Alejandro Campo in 2016 that started the entire partial reprogramming field, who said that based on the study, with careful modulation, aging might be reversed. And I think partial reprogramming could be just one way that could help us achieve this goal of reversing aging. So with that, thank you very much. And yeah, if you have any questions, happy to take them.
Okay, awesome stuff. Thanks, Yuri. Okay, we have time for just one question.
Oops, I have to. No, I'll make it quick. So in terms of transient therapies, are we still looking at doing transient iterative treatments, or are we looking at doing a single permanent treatment? And the second question, in terms of gene delivery, would this be a good option for PLVs with cell-specific ligands?
I need to think about the second question, but the first question, the way we do it, it's a permanent therapy in terms of delivering our cargo, which is inducible, and until you induce it, the cargo is not expressed. So from that standpoint, this therapy, if it somehow turns out to not be as safe as we think, if you just stop expressing those factors, stop inducing this therapy, we expect that you can then just avoid any potential side effects. But from our standpoint, because we think partial reprogramming needs to be applied on a periodic basis, we need to deliver a therapy that will stay there and can then be activated when we need to activate it. So from that standpoint, it's permanent.
You don't need to put in a kill switch?
Well, since it's inducible, like if you don't induce it, that's already the kill switch. So from that standpoint, we don't think we need to do anything else, like excise it or anything like that, yeah.
Okay, great. Let's give another round of applause for Yuri. Thanks.
Thank you.
Thank you, Yuri.